Hi, I'm Kevin Moore. I'm the founder and CEO of Quilt.bio, and I'm here today with Vasisht Tadigotla to talk to you about accelerating cross-assay correlation with GenAI. Vasisht, over to you.
Hi, I'm Vasisht Tadigotla. I run the data engineering team at Biomedicines, which is a biomedical company with circular RNA, and happy to work with Quilt to make sense of our assay data that's scattered across multiple locations and places. So one of the big issues that we have with data in general is we have multiple processes and we have multiple systems that collect data. For instance, we have NGS data that we process as Nextflow that goes into Quilt. It's nicely packaged with metadata, experimental information, etc. But then we also have on the same samples, we have other assays that are run that are low throughput, let's say ELISAs, PCRs, things of that nature. Flow is another example. So now you have two separate systems with data in two separate databases, for, if you will. And it makes it a little harder for teams to be able to draw inferences across the data set. So if you want to correlate your NGS expression with whether it's RNA-seq or mutational profiling or single-cell sequencing against protein expression data or flow data, it becomes a little challenging for both for our end users and for our data scientists and bioinformaticians because they have to query multiple locations to pull data in, combine the data sets together, do the analysis, generate a report, and distribute it to our end users, which are typically folks that are in the lab. Next slide, please, Kevin. So a lot of the time is spent trying to figure out where data is, right? So it's a simple question. I have a set of compounds that I'm testing against samples, and I want to understand, in this case, let's say, which of these compounds upregulate the NF-kappa B pathway. So in order to do this, first you have to pull in all of the expression data from Quilt. Then you have to match the metadata with the results that are in Benchling, match the samples against each other, then pull in all the files. So it's, you pull in from the warehouse, you pull in from unstructured data, then do a comparison, generate maybe a Jupyter notebook of sorts, or perhaps even a Quilt package. And that's how you present the data. But all this process takes time. And for an experimental person who's doing these experiments, they would want to know, did my experiment work? Did this compound work? What are the experiments that I need to do next in order to either verify my hypothesis or generate a new hypothesis to test against? So that becomes challenging in doing it in this form and fashion. Kevin, on to you.
Fantastic. So for those of you who are not familiar, Quilt is a scientific data management system for AWS. We give an intuitive and reliable interface to storage resources in AWS, particularly Amazon S3. We give you repeatable we make your workflows repeatable with data versioning. We help you govern that and provide access control and monitoring. And we do this all inside your AWS account. Previously, I've spoken with colleagues here at the Nextflow Summit about how we can solve some of the problems that this just was mentioning using tools like federated querying and particularly with SQL to replace that file wrangling with simple queries. And so if any of you were here or were in Barcelona last year for the talk, we were looking at questions like which cancer types in CCLE have the highest expression of EGFR and mapping that to SQL queries. But as we talked to users, we realized that that still took a lot of work. We had to set up the federated querying. It still produced a dependence on users knowing SQL, maybe users plus a little help from ChatGPT. But now it's 2025, and so we really are thinking, what if we don't know? So can't AI just do this for us? And happily, the answer is yes. But to make it work, your AI needs some tools. There's been incredible development over the last year in AI systems, and one of the most exciting is the adoption of what's called the Model Context Protocol. This is the standard for adding capabilities, or as they would call them, tools, to your AI systems. MCP servers enable large language models like Claude or ChatGPT or even Cursor to pull information from multiple sources and even to actually take actions on your behalf. thinking about the use case that we mentioned here for cross-assay correlation and more generally for bioinformatics data analysis, there's a couple of essential tools that we've identified to really accelerate bioinformatics workloads. First of all, a lot of you out there are using the cloud, using cloud object stores like Amazon S3. So the ability to search and query S3 to find relevant data sets in S3 quickly amongst the hundreds of terabytes of data and millions to billions of files there. Also to read those files, determine their structure and schema, and then to a scale out SQL query interface so that we can take advantage of the SQL efficiencies that we mentioned in our talk last year. Also, though, not all that data is in Amazon S3. A lot of that data, as Vasisht was mentioning, lives in other systems. The ELN and LLMs systems house a lot of that important metadata, particularly related to samples. So the ability to read sample metadata, to query things like results tables, and to collate and analyze data from your LLMs is really important to rounding out this analysis. And finally, once that data is pulled together, the ability to create data visualizations, which will communicate those results to both the analysts themselves and to their colleagues in a way that's interactive and understandable, and then to have a platform to host those visualizations and share them with the team. In the particular example we've drawn today, we're looking at two MCP servers connected to a large language model, Claude Sonnet. In this case, we have an MCP server under development from Benchling that can read sample data, query results tables, collect and analyze that data. And then an MCP server recently released from Quilt that will search and query S3, finding the relevant data sets, and help build visualizations into Quilt packages, and also share not only the results of the visualizations themselves, but package together in a version of reproducible way the outputs of those queries. Jace, can you take us through the specific example for protein abundance?
Yeah, so this is a fairly common issue that we face in our day-to-day work. We have expression from RNA-seq data, and we want to know how well does this correlate with the actual protein expression. So you have the transcription data, how well does things translate? Because at the end of the day, we're interested in the protein expression. So our RNA-seq data is typically analyzed using Nextflow. We generate Quilt packages with Nextflow, and we also have a tabulator table set up for the salmon gene abundances. And all of our experiments, which are low throughput, such as ELISA and qPCR, et cetera, are tracked within Benchling. So we have registration tables for registering the samples. We have results tables that assign results against these samples, and those are all stored in the Benchling warehouse, which is a Postgres-based database. So the typical process for this would be someone from the bioinformatics team or data science team would get a request saying, oh, we've done X number of samples. We would want to know for these samples, what does the expression of genes XYZ, does that correlate with what we find in protein expression via ELISA? And that process usually tends to take a few days. People have to collect data, merge things, ask around in terms of where the metadata is. And despite best effort, sometimes the sample annotation is not always there. So there's multiple questions around that. Is the annotation in Quilt? Is the annotation in Benchling? So what we have as an example here using Claude and the MCPs is, can we connect the data from which is in Tabulator in AWS with the Benchling dataset? And can we enable perhaps a bench scientist to do an initial analysis that can answer these questions for them? And they can then take that analysis to someone who's in a data science or a bioinformatician and do a deeper dive to understand the ins and outs of the data, what are the next experiments to be done, and potentially could even Claude suggest next experiments based on what it infers from the analysis of the data. So now let's look at how we compare data using MCPs from Quilt and Benchling. So the query here is we have a bunch of Quilt packages that are in a specific bucket. There's tabulator tables, which are tables that parse the salmon data that are associated with it. But in the interest of time, we reduced the sort of the number of genes that we're looking at to a shorter subset and a few samples that are representative of the study. And then we're asking Claude here to use the MCP to pull the data from RNA-seq expression data from the Quilt samples, and then compare this data to the samples that are in benchling. And once that's done, we're going to create a Quilt package and also build some visualizations to see what are the differences, what are the similarities. So let's see what it does. So now we have the query set up. It's going to think about it a little bit, and then we're gonna go start querying the Quilt bucket from the NCP side. So it's looking at how do we pull the data from the study data. We use an Athena query. And then once the data is done, it's gonna initiate the benchling MCP, which is called benchling research. So now it's pull out the query for the tabulator. Let's give it a couple of seconds. Move it along here a little bit. Now it's pulling in the benchling data. So this takes a little while. So we've skipped through some of the steps. Now it has the data from benchling research. It's pulled in the expression data. It's compared the sample IDs between benchling and Quilt and the gene IDs between benchling and Quilt. And it's comparing the data for samples S to the S10. And now it's creating an artifact for the expression data comparison. So now it has pulled in data from the S3 bucket and pulled in data from the benchling warehouse and plotting out the different attributes that we want. Mostly for the genes that are expressed in RNA-seq, do we see consistent expression in the protein expression data? Or are there any outliers one way or the other? This is analysis that's fairly common. You have multi-omics data. So this is sort of something that people want to do on a regular basis. So now that we have all of these plots that show the distribution of the samples, what are the key patterns? In some cases, you have RNA-seq data being high, but no protein expression data, and vice versa in other cases. So these are things that the experimental scientists can take further and see, are these because of certain conditions within the experiment? Is it because of the biological considerations that we need to look at the next experiment? So now we have the analysis. It's on someone's desktop. They're looking at it, but then the Quilt MCP will take this analysis, package it up, and create a new Quilt package with the analysis. And that package can now be shared with other bench scientists or data scientists, and they can then look at it, use it as a point of discussion. And this whole thing takes about 10 minutes, and there's no wrangling of files, there's no SQL queries, you don't have to ask anyone to help you build a visualization. It's all done within the Claude desktop application. So this enables scientists to be able to ask fairly deep questions about the data that they've generated from multiple different studies across multiple different platforms. In this case, we're using benchling and Quilt as an example, but you could potentially have data in other Postgres databases that you could use a Postgres MCP, or you could use something else that you have an internal MCP that you've built out for data that's very specific to your company. So as such, we think that this is a good process in terms of how best to democratize the analysis so that the bench scientists can see the fruits of their work without having to have everything run through the data science team. So again, this is an initial analysis. I'm sure we can do deeper sets of analysis either by asking Claude to query or chat about what the findings, or take this analysis to a computational biologist or bioinformatician to further dig into why and where and what sort of questions should be asked. So at a broad level, this helps speed up experimentation in our specific use case. So now that it says it's created the package in Claude, let's see if it's actually done it. So let's go back to the Quilt package. Now you see that it's created a Quilt summary. It has created separate visualizations for gene expression and for protein expression. This is the protein expression plot. We can also go back to the specific plots for gene expression and protein expression, which are JSON files that you can replot as charts. And you see the protein expression for those samples, CXCL10, IL10, and you can go back and look at the equivalent RNA expression. And there you have it. 10 minutes from querying the data set to getting analysis that is useful, at least as a starting point for the next steps. So the takeaways from the demo is you can quickly do analysis of multi-location data, whether it's in Benchling or in Quilt or in S3, without having to figure out what SQL queries to generate, without having to download any files. And being able to provide this information quickly to the folks in the lab makes it easier for them to iterate on experiments. And the analysis teams can spend more time on newer ideas and more complex analysis than the basic questions around, did my experiment work? Or is this my favorite pathway upregulated? Or what's the difference between expression of genes A, B, and C in this experiment versus the experiment that was done last week? So these are questions that are fairly common, but take long time to answer, even with custom dashboards and analysis tools. So being able to provide this information directly to a lab scientist without having to spend time working with the data science team, I think is a helpful thing to have for us, particularly given that we are a small company with not a ton of bandwidth within bioinformatics and data analysis.
Vasisht, thanks for that. Following up, this has been an amazingly exciting year in the development of AI tools. Model Context Protocol is an incredible new development that allows us to work really in a new way from what we had done before. I think we're all used to, by now, working with systems like ChatGPT, and I know that almost all the software tools that I interface now have their own chat interface. Model Context Protocol and MCP servers gives us a new way to add AI or to use AI to power our workflows. Now we can coalesce capabilities and information from multiple systems, in this case, Benchling and S3 via Quilt, into one session with our AI interface of choice and give us access across systems and really unlock the potential of this amazing new technology. If you have any questions for either of us, we'll be in the Q&A session, and you can reach us by email at the emails on the slides. Thank you.